So good afternoon. Welcome to the Philadelphia Regional Center for Children's Environmental Health, PRCCEH seminar series. And I am Aimin Chen, co-director of the center, together with Dr. Rebecca Simmons and Dr. Marilyn Howers. So we have a couple of running slides before we introduce speaker. So Jessica, yeah. So I just want to acknowledge the land of University of Pennsylvania is on the land of Lenape. So it's my great pleasure to introduce Dr. Hannah Choi for today's seminar. Dr. Choi is come here from Lehigh University. She completed her PhD in environmental health at Columbia University and subsequently postdoctoral training at Harvard School of Public Health. She is currently associate professor and director of the Children's Environmental Precision Health Institute at the Lehigh College of Health. Her research interest is mainly to use multi-scale network biology or multi-omics approach to understand asthma etiology as well as mechanisms during early life. She has studied polycyclic aromatic hydrocarbons, PAHs, particularly the benzoylpyrene, BAP, and moisture exposure and volatile organic compounds, VOCs, and child asthma risk. So she has done study in multiple countries, including Czech Republic, Poland, and the United States. And also she examines outcomes like birth outcomes and child obesity, also difference in boys and girls. So she's a member of the PRCCEH and actively involved in research translation to policy making as well as to community members. Today, she will present her insights on air pollution and asthma with the title, Does Air Pollution Contribute to Unique Asthma Subtypes?
So I've been digging at this question of, are there subspecies of asthma? Specifically, are there specific diseases that's associated with air pollution? So I will briefly lay out the background and the context as to how I came to pose this question, introduce the personnel that contributed to this data, and discuss the implications very briefly. So I have to introduce, this is a very descriptive data analysis by Impressionist painter Monet, who in the turn of the century, at the height of the Industrial Revolution, made this observation that in spite of the fact that we are looking at a sun that is at the height in the middle of the day in the moon, in the middle of the sky, we're seeing an overall incredibly dark screen that due to this intensive burning of fossil fuel in the at the height of Industrial Revolution. So I like to introduce that the pollutants that we'll be talking about today are these ubiquitously distributed, well-known pro-mutagen and pro-carcinogen that we inhale, everyone inhales all throughout our lives, all over the world. And in particular, these are, if you look at these are generated from cars, factories, cigarette smoke when we cook barbecue, and they are so adept at infiltrating through our, skin, blood vessels. And if you look at the picture on the lower left, they really stably latch themselves on DNAs and induce a whole array of health effects, including reduction in birth weight and really impaired development of placenta, as you see on the lower right-hand corner, where figure C represents the cigarette smoke exposed placenta that looks overall sparse and very blue, reflecting poor oxygen innervation. So why do we need to think about asthma species? Could tell you the bottom line first. It's essentially that we've been digging at this question for the last 4 decades, but we do not have a clear understanding as to what we're dealing with. There's absolutely no cure, thus there's no prevention to speak of. And by a very conservative estimate, $56 billion in US dollars per year is actually a very antiquated estimate. It's more like $86 billion per year in US alone, as of 2016, I believe. And in about 38% of children, one and the only intervention therapy, that is the eosinophil suppressing treatment, is ineffective in about 38% of children who are expected to grow up to develop severe asthma cases. So, extensive review of the literature, told me that the the current approach of considering one exposure pollutant, looking at one gene, one molecule at a time, and looking at the outcome, perhaps is a very woefully inadequate way to tackle that complex behemoth called asthma, but rather that by deciphering the mechanism as the basis of defining asthma subdiseases, we can tailor the clinical, public health, and individual level intervention approaches much more thoughtfully. So figure on the left shows the temporal explosion of asthma prevalence in multiple countries since 1965 and kind of leveling off in more recent decades. So in 2014, NHLBI convened and made a recommendation that we need indeed a new way to research asthma. That is, they recommended that we need developmental origins of asthma development, and that we need to go beyond looking at the phenotypes to understand the mechanism-based basis of disease development, which is so-called endotype. And then finally, in doing so, clarify the association between the mechanism as the basis for defining the endotype as well as the phenotype groups. So in essence, the goal of this work, my work, has been on integrating multiple omics, which in hindsight right now is a bit outdated, it's getting old, and to use that as the basis to identify at least 2 subtypes of asthma, type 2 and non-type 2. So how is this different from how we've been thinking about this? It is a little bit different because we are conceptualizing that genes are fluid entities which responds to specific environmental context. So in a homogeneous population, when the children in very early life get exposed to very well characterized environmental variability, we could go beyond that notion of GWAS, which postulated that we will identify these very high penetrant and highly expressive gene that's shown in this red diamond. I don't know whether you could see here, would identify the genes that are highly predictive of asthma. Oops, sorry. But that turned out to be a huge disappointment. So much more recent development is that by identifying the networks of genes as shown in gene-gene interaction in the middle top and bottom figures, where we sequence the DNA variation as well as RNA abundance, we correlate that in conjunction with the exposure as well as the phenotypic outcome to get at the understanding of the relationship of the groups of much more weakly acting gene groups that work together to contribute to the development of asthma. The way of the future that we like to argue is that by characterizing the DNA variation in conjunction with much richer array of multiple scales of RNA, protein, metabolite data in conjunction with environmental exposure as well as the phenotype, we get a much more comprehensive understanding of gene regulatory network that control these multiple layers of molecules. So the goal of this is to see whether we can postulate asthma as specific subdiseases that result from host susceptibility in conjunction with the childhood exposure to environmental air pollution exposure, and in the process to elucidate the mechanisms. So the first goal was to propose the mechanisms using a case control study, using an overall brute force approach, where we postulate benzoylpyrene, the well-known carcinogen. These have been shown to be direct carcinogens in multiple species and for multiple decades. Whether they induce not only the dichotomous outcome of asthma, but also the ordinal outcome of asthma severity in what I termed AMOD index, asthma severity and diversity in the index, and whether that's mediated by modular gene co-expression networks. And then we wanted to ask, who are these kids? Can we characterize them in terms of their prominent clinical features as well as biomarker characteristics to kind of correlate whether their asthma subtypes correlate with prominent features in terms of the asthma subtypes? So the methods. So we lucked out in the sense that Czech population is overall homogeneous. They're overall non-stationary. And so we enrolled children in two areas in this Ostrava region, where for multiple centuries, they've been a highly industrial region right next to Poland. We also selected children from this very rural, clean area right above Austria and stratified them according to cases and controls in these two regions. We also obtained very rich environmental data in conjunction with the DNA methylation and whole genome transcriptome data, and analyzed them through pretty much brute force approach, and conducted an integrative network analysis in which the cases and control children defined as A1 and A0 from the rural Czech Republic and industrial Czech Republic area were analyzed in terms of their DNA methylation and whole genome transcriptome. In terms of these very, millions of sub-analysis where we correlate the degree of correlation between methylation with transcription, methylation with clinical traits, methylations, as well as transcriptome with the benzoylpyrene levels. We sent them back to see whether the correlation patterns tell a cohesive network of genes shown on the C, and whether such groups of modules we call them, are consistent with what we know to be a known pathways in the integrative network analysis. We then identified whether such dysregulated gene modules represent certain well-known groups of modules, and then also rank them kind of to create a certain structure, hierarchy. So if you were to see these genes as this gang network of gangs, to see how the hierarchy exists within such gang groups. So to kind of rehash, I mean, these were specific hypotheses that we posed, whether exposure is correlated with the epigenome, whether the exposure is correlated with the transcriptome, and all collectively, whether they tell a cohesive story in terms of the outcome. Well, actually, I could come back to this later, but essentially, in the urban, the children that came from the industrial region were exposed to significantly higher levels of air pollution, which is shown on the y-axis, and that the severity of asthma was significantly and in a linear fashion was associated with the severity of asthma in both the non-atopic children as well as the atopic children compared to the control children, but only in the industrial residing groups of children. So here's the bottom line, and I apologize that it's extremely busy and very difficult to tell, but I will break this down for you. So what does this mean? So I just wanted to show you a network, or more or less an archipelago of genes that collectively contribute to the outcome. Conceptualized in terms of the asthma severity and diversity index. And what does each of these modules mean? It's specific groups of the pathways that collectively contribute to the pathogenesis, what we like to think of as the pathogenesis of asthma. And if we were to zoom into individual ones, again, I mean, I apologize that this is extremely busy, but to kind of decipher them, each lines on the top dendrogram shows you each child, both the cases and the controls. And each line would be comprised of the relative abundance of cell population, immune cell population. Oops, sorry, I didn't mean to get ahead of myself. And the dendrogram below that would be the severity of the expression of the asthma cases and the controls, and more the ordinal outcomes that's been expressed. Below that, we have the level of intensity of exposure variable, air pollution intensity, as well as the marker of cigarette smoke exposure, cotinine. So what are we actually seeing? What we're seeing is that the severity of asthma outcome, even though it's very difficult to tell in terms through a human eye, the machine learning technology was able to discern that severe asthma outcome was associated with the distinct subgroups of lymphocytes. What are they? So this is actually kind of the bottom line of gene expression that correlates with the immune cells, where the y-axis would correspond to levels of degree of either the activation or expression, elevated expression or depressed expression of each of these gene modules. And the x-axis would be the specific cell types, immune cell types. And what we are seeing is that that very distinct groups of cell types are significantly correlated not only with the air pollution exposure, but also with the degree of gene expressions and the epigenetic regulation of gene expressions. So what are they? So the top 2 genes that were identified were both focused on suppressing the nuclear factor kappa beta pathways. So these are the activation of nuclear factor kappa beta pathway indicates a pretty severe asthma. And both of these genes were focused either activated to either suppress the nuclear factor kappa beta gene, or in the case of pro-inflammatory gene that, oh, sorry, it's very sensitive to my touch. In the case of the bottom right figure, GFAN-D6, the pro-inflammatory gene that elicits nuclear factor kappa beta pathway was suppressed, so that ultimately nuclear factor kappa beta pathway would be suppressed. On top of that, we identified that benzoylpyrene also drives the heme biosynthesis, which promotes the activation of iron transport, maturation of immature neutrophils, as well as the increase in mature red blood cells, erythroid cells that would become red blood cells. I will get back to what this means in the later slides, so bear with me for just 1 second. So collectively, this is the bottom line. This is the linkage between gene expression with the cells that result from that, and also the cell types that result from the dysregulated elevated gene expression or depressed gene expressions. And what story are they telling? Essentially, well, let me actually skip to the figure that explains that story. What is becoming very, very clear in the last 5 years is that air pollution that's not limited to benzoylpyrene, but benzoylpyrene is certainly one of the pollutants that induce this, disrupts the normal ion homeostasis that we maintain within our system. So this is actually not, it's a finding that's come into light in the last 5 years. And such disruption in ion homeostasis results in what's known as ferroptosis. And this is a mechanism that's distinct from apoptosis. And it results in upregulated oxidative stress in people who experience disruption in ion regulation. And such increase in oxidative stress, altered signal pathways has been associated with the neutrophilic asthma. So the pathway that you have just witnessed is associated with the elevated neutrophilic or non-type 2 asthma in children that predominantly reside in the industrial Czech Republic regions. So to backtrack a bit, the goal of the second aim was to identify the kind of the prominent features of these children in terms of the biomarkers, so that we have a bit of a handle on like the point of entry, so to speak, as to what the prominent endotype biomarkers should be. So to make the long story short, I stratified the children coming from both the rural Czech Republic as well as the industrial Czech Republic, and stratified them, put them all together and stratified them in terms of their allergic atopy status, where the children on the left are predominantly non-allergic children. Further stratified in terms of sex, boys and girls, and then further stratified in terms of their lung function. On the x-axis, x-axis shows the controls and cases, where the controls and case children are stratified in terms of their obesity and overweight status. So we have 4 comparison groups in contrast, even though we have cases and controls. So leftmost group would be the lean controlled children, followed by overweight, obese controlled children, followed by lean asthmatic children, followed by obese and overweight asthmatic children. In boys alone, on the leftmost panel, and we see that the children who have the highest median exposure to benzoyl-pyrene, that is 20 nanogram per cubic meters of benzoyl-pyrene, groups are associated with the lean asthma. In contrast, among the non-atopic children, the girls that have not only the lean asthma, but also the overweight, obese asthma, and very severely impaired lung function, were associated with the highest median levels of benzoyl-pyrene. In the allergic boys, we don't see any clear trend in terms of the obese asthma status and the benzoyl-pyrene, median benzoyl-pyrene exposures for the lung function impaired or non-impaired children, but only in the obese asthmatic girls, we see that the lung function impaired girls are again associated with the highest levels of benzoyl-pyrene. When we stratify the same groups of children in terms of their lipid oxidative stress level, we see something really even more intriguing. Only among non-atopic children we see, and in girls, so I'm going to start with the group that shows the clearest trend. Among non-allergic children who happen to be girls, we see a linear dose response relationship in which those girls that have the higher than the median oxidative stress level are also associated with the highest median benzoyl-pyrene. In contrast, it's the lean asthmatic boys who are also non-atopic, who also have the highest oxidative stress level that's associated with the highest median benzoyl-pyrene concentration. We see something completely different in the allergic boys. We don't see any trend among allergic boys, that is in terms of benzoyl-pyrene exposure and obese, overweight asthma status. But among the girls, we see something of a much more muted response where it's the lower oxidative stress experiencing girls who also happen to be who also happen to be overweight and obese, that happens to have the highest association with the median benzoate pyrene concentration. So I love drawing lines. You know, I drive my students crazy by asking them about shape of the dose response all the time among the undergrads. So I had to, in spite of the fact that this is overall very small sample size, had to draw a line to see whether we are seeing any intelligible trend here. And what's notable here is that among the lean children, we don't see much of a trend, but among the overweight and obese children who happen to be non-allergic, who also happen to be girls, there's a inverse association between increasing benzoate pyrene exposure and reduction in lipid oxidative stress level. But the sample sizes is severely limited. And among the allergic girls, we don't see any trend, or boys. What happens when we do the same analysis looking at the DNA oxidative stress? The trend is overall similar, but much more muted. So only in the group that actually has low levels of oxidative stress, we actually see a stronger linear correlation with the ambient concentration of benzoate pyrene among non-atopic girls, going from lean control to overweight control to lean asthmatic to overweight asthmatic asthmatic girls, sorry. And consistent with the prior analysis, only among lean boys, we see an elevated oxidative stress level associated with the elevated ambient benzoate pyrene concentration. Again, we don't see any trend among allergic boys that are also again stratified similarly, but among the girls that have that are allergic and have a lean asthma or obesity associated asthma, we see a linear increase in benzoate pyrene concentration. So if I have thoroughly confused you, and I apologize, but when I put all of these into the model, we believe that in spite of the fact that sample size is overall limited, we see overall, I mean, really potent association posed by benzoate pyrene on both the lean asthma as well as overweight obese asthma after we account for the oxidative stress among the non-atopic asthmatic children. We don't see similar association at all on allergic boys. Benzoate pyrene has no risk, poses no risk on allergic asthma, but only among girls, benzoate pyrene is associated with a significantly elevated overweight obese asthma after accounting for both the lipid and DNA oxidative stress. So this is the bottom line, that we see something about divergent association between non-allergic and allergic asthma, where allergic asthma within non-allergic children, benzoylpyrene is associated with 10 to 20 times greater odds of either lean or overweight obese asthma. And benzoylpyrene is associated with a significant asthma risk among, is not associated asthma risk among lean atopic children. We see strong effect of both the lipid oxidative stress and DNA oxidative stress, perhaps mediating the non-atopic overweight obese asthma. But the association is much, much weaker, even though it's significant, among atopic overweight obese asthma. So overall, collectively, they point to overweight obese asthma being associated with more severe phenotype, and that that it's collectively associated with the higher median benzoylpyrene concentration in both the non-atopic and atopic children. So I explained this slide, and overall, we believe that benzoylpyrene is associated with ferroptosis mechanism, particularly among the non-atopic children. But this is pending further validation and replication. So this study has a number of strengths and limitations. So I'm going to go through the limitations first. Most notably, this is admittedly a case control design and is limited in terms of its etiological attributions. And the sample size is limited. And that we use for the simplicity sake, we consider benzoylpyrene as the marker of fossil fuel related pollution level. However, this is supported by a number of other research that has shown very powerful correlation between benzoylpyrene with other fossil fuel generated burning related pollutants. We did not collect any personal air monitoring due to limited budget. Then the strength, we kind of looked out that Czech population is overall very stationary and that they are overall genomically very homogeneous. So we believe that the environmental differences are stark in contrast. And it's not just our analysis, but number of growing body of other data that supports the benzoylpyrene and DNA adduct as the inducer of ferroptosis mechanism, most notably in rat and mice models that are coming out in the last 5 years. But our analysis shows for the first time that ferroptosis might be a driver of severe non-eosinophilic asthma. And that we are seeing such outcome most severely in non-allergic, overweight, obese girls who are also experiencing severe oxidative stress suppressions. So what are the unknown unknowns? This is only the beginning, and I mean, benzoylpyrene has been associated with a whole host of other outcomes, some of which are named here. And I thought this is really a nice point of introducing the beginning as to how we might benefit by collaboration going forward. So I will open the floor for any comments and questions at this point.
Thanks. This is a great talk. And I think, people could open up and ask questions. You don't have to type in, you just like unmute and speak yourselves. Marilyn first.
Hi, thank you so much. What a fascinating talk. I just love that. I was thinking about the exposure measurements and wondering whether some of the differences between the genders, boys and girls, might have to do with the fact that some of the exposure may not be just from the air. The environment that you described is one that would have put lots of legacy PAHs and benzoate in the soil. It's a relatively hydrophobic material and tends to stay where it is put. And so I was just wondering, there may be a differential in terms of the contact, amount of time, and disruption of the soil between boys and girls, especially over time. And I was wondering if you thought that might be playing a role.
That's a really, really thoughtful observation, and thank you so much for raising that. We tried our best to characterize the behavioral traits of the teenagers and pre-adolescent children that were enrolled in our study. But because our sample size was so limited, we are always at the same time worried that we might be torturing the data beyond what it's capable of telling us. So it's a really fascinating question that we would love to examine further. Do boys tend to play outside more and girls do other things indoors more? I would guess that, but I do not know for certain. So this indicates how important it is that we work very closely with the people that contribute to the study. Yeah.
Thank you.
Okay, I do have a question. So I think the talk is fascinating. Basically, we have to think about atopic status, weight status, obese versus slim. And we need to think about boys and girls. So but actually for the BAP levels, benzopyrene, I was thinking if we want to do a similar study in the US, how was our BAP levels compared with Czech Republic? Do we actually, our levels are too low that we won't be able to find anything that is clearly, or is it we still actually can go down the lower exposure levels, we can still find something? This is just a hypothesis to think, and how low of the exposure this could be, could be studying basically.
To venture, I guess, the preliminary data on benzoate-pyrene in the US tend to be a whole lot lower than that in Eastern Europe. In spite of the fact that Eastern Europe as a whole, are like turning away from fossil fuel burning in many countries. But having said that, I am also struck by how Pennsylvania, in different pockets of Pennsylvania, we still have for whatever reason, we have these, what I like to think of as hills and valleys that seem to capture and not let out these pollutant concentrations that I would carefully venture a guess that in spite of the fact that we may see annual averages that are about 5 to 10 times lower than that in Czech Republic, that may be a poor sure indicator of actually what's biologically and health relevant, that if we were to do a personalized, miniaturized exposure assessment, our sensitivity might be much higher and might allow us to capture health outcome relevant exposures still. So I feel very brave that we happen to live in this very typical Rust Belt region where burden from asthma is just so unimaginably high.
Yes, yes, we have 20-some% prevalence in the urban children. That's very high. And as Marilyn mentioned, we not only exposed to really air pollution for benzopyrene, barbecues, some other exposure sources. Okay, so even the traffic. Okay, so the fossil fuel burning. Anyway, so this could be some other actually potential exposures.
Well, thank you so much.